Good morning everyone. I am Harita, DL Botany at Telangana Tribal Welfare Residential College for Men, Sangareddy. In continuation with the previous session that was on 7th, I would like to take another topic from physiology, that is regarding plant transpiration. Now, before I start with my presentation, I would like to put before you a few observations. Imagine that you are walking on a midsummer afternoon and you see a very large tree and move underneath it for shade. Imagine that you are moving from outside into a large garden which has a collection of large trees, plants, flowering plants, etc., etc. Imagine yourself walking into a crop field, a rice field or a maize field. What do you observe? What do you feel when you move from hot sun into shade of a plant, when you enter into a garden or when you walk through a crop field? You will start feeling cold. Why do you think this is happening? Is this the environmental conditions that are leading to that coldness, or is your body reacting to it, or what is it? This is one of the physiological processes that is occurring in the plant, which is correlated with another two major physiological processes of the plant, that is transpiration. Today we are going to deal with the plant transpiration process, and now on the slide you can see I have put atmosphere, plant and soil and interconnected them. Just observe them and try to infer. Now, for a plant, since it is a living organism, its components, whether it may be cell components or the organism itself, is made up of 99% of water. So, water is a very important essential feature for the plant for its survival, and not only that, for absorption of water, along with the water, important minerals are also absorbed by the plant. Now, what is the source of this absorption? Soil. So, through its roots, it absorbs the required amount of water as well as essential minerals and takes it up and passes on to the aerial parts of the plant. Now, after taking up the required amount of water and utilizes it, you will find that the extra amount that is taken up by the plant is given out in the form of water vapor, and this water vapor enters into the atmosphere. You will be surprised to know that the atmospheric water, the total content of atmospheric water, nearly 15% of the atmospheric water is contributed by this evapotranspiration method. Now, look into this slide. Now, here you can see that a large tree rooted into the ground, and we find that the roots are the source of absorption, as I just told you. Now, once the absorption continues all through the day, whatever need of the plant is, that amount of water the plant absorbs. You will be very surprised to know that out of total content of the water that the plant has absorbed, it only utilizes 1% of it to perform its various metabolic activities, and the remaining 99% moves out of the plant through the leaves by transpiration. So, all the excess water or 99% of the water gets evaporated by this process. And how does this process occurs? Because not only on soft aerial parts of the plant, specifically on the leaves, you find that small tiny pores are present, openings are present, called as stomata. Through this medium, this excess water escapes into the atmosphere in the form of water vapor. So, let us move on further now. Now, many physiologists conditionally use this term that transpiration is a necessary evil. Why do you think this is? Because I have just told you that out of total amount of water that a plant absorbs, 99% goes out of the plant does not utilize it. Then what is the usage of taking up so much of water? Now, this is because of root pressure and the other related metabolic processes. But transpiration at such a high rate, how does a plant get benefited? One thing is that it can take out the excess amount of water from its body, that is one thing. Second is that the transpiration helps in keeping its surroundings cool. So, it cannot escape this process called as transpiration. So, you find that, as I have just told you, this transpiration process, you find that depending upon the kind of species, the amount of water that is lost through this process called transpiration can be thousands and thousands of liters. Okay? That is why it is called as a necessary evil. Now, let us look into the definition of transpiration. If you are looking at the definition of transpiration, it is defined as a process in which excess water leaves the plant through in the form of water vapor. Okay. And what are the chief organs for this transpiration to occur? It is primarily the leaves. So, since the leaves are flat appendages and maximum amount of stomata are found on the leaves, we find that leaves are the primary source for transpiration. Besides that, we can also find a good amount of stomata depending upon the plant species and depending upon the where this particular plant grows, whether terrestrial or aquatic. If it is terrestrial, whether it is a temperate plant or a tropical plant or otherwise. So, we find that the number of stomata will vary, and on the soft aerial parts of the plant also, like on stem, on floral parts and on fruits also, you come across varying number of stomata. One more thing I would like to add is that now in most of the plants, if you observe closely, you find a thin waxy coating over the surface of the leaf. Now, this is called as the cuticle, that is the outermost protective layer beneath. After it, we come across the epidermis. So, this cuticle is made up of a deposition called as the cutin. Now, how this cuticle is important? Cuticle can be thin or thick. So, if we find that the cuticle is thin, then transpiration occurs through the cuticle also, that is called as cuticular transpiration, that we will deal in the next slide. And later, if the cuticle is thick, we find that it suppresses the transpiration or interferes with the rate of transpiration. Not only that, it is amazing to know that if the cuticle is completely absent on the leaf, then leaf will have a deleterious effect, because water will continuously keep on evaporating, because there is no protection layer or no waxy layer over it. Okay. Now, let us go further. So, as I told, depending upon from where the transpiration is taking place, we can divide the transpiration process into three categories, that is stomatal transpiration, cuticular transpiration and lenticular transpiration. So, not elaborating much on them, stomatal transpiration is a transpiration occurring through the stomata, whereas cuticular transpiration is the transpiration occurring through the cuticle. Now, the cuticle transpiration is nearly 5 to 10% of the total transpiration. So, 90% of the transpiration occurs through the stomata. So, remaining percentage occurs through the cuticle. Okay. Now, if you look into large woody trees which usually shed their leaves during the winter season, what we observe that these large woody trees, even they need to breathe. So, they have small openings on the woody barks or woody stems, which we call them as the lenticels. So, these are the places where gaseous exchange takes place. Now, one more thing that I would like to add over here is, when we are talking about transpiration, it is not only evaporation of the water in the form of water vapor and excess of water moving out of the plant. Besides that, it is also the point of exchange of gases, that is CO2 and O2. We know that CO2 is the primary one in photosynthesis. So, it will the path of CO2 from the external medium into the plant and moving out of oxygen from the plant after photosynthesis outside is through the stomata. So, transpiration based upon where they occur and how they perform, three types are there: stomatal, cuticular and lenticular transpiration. Now, let us learn few more facts regarding the transpiration. Now, most of the stomata, by general rule, they exhibit diurnal patterns. That is, if you take normal plants or mesophytic plants or large group of plants, normal plants into consideration, we find that stomata open by day and close by night. That could be interrelated with the photosynthesis, and of course, photosynthesis is once again dependent upon the sunlight. So, light is one of the triggering factors interrelated with the other factors also, like temperature is there, water is there. These all work in tandem in opening and the closing of the stomata. So, these even the stomata exhibit this diurnal pattern, because most of the leaves exhibit this one. Okay. So, what do we find is that in early mornings the rate of the transpiration is quite low, but as it approaches afternoon, that is mid afternoon, the rate of the transpiration gradually increases, and after early evenings we find that there is a constant decrease in the rate of transpiration, and by night the transpiration rate is quite low. So, that is the inference. Now, let us move about it. Further we will elaborate depending upon the nature time of opening and the closing of stomata, Loftfield has classified stomata into 3 types. What are those type? Alpha alpha type, potato type and the barley type. If you look into the first one, alpha alpha type, now what do we see is that stomata are generally open all through the day, but they close after mid afternoon for some period of time. After just before the sunset, once again these stomata once again open. Okay. So, we find the closing of stomata only during the mid afternoon time, rather than otherwise they are open all through the day. Example for such kind of stomata, we can see them in pea, apple and other thin leaved mesophytes. You come across this kind of stomatal type. Okay. If you look into the second type, that is the potato type, what do we see that this stomata is closed for 3 hours after sunset, otherwise they are open all through the day. Such kind of stomatal opening is termed as potato type, and we usually see them in most of the vegetable and fruit plants like onion, banana, pumpkin, cabbage. You find such kind of stomatal pattern. Okay. If you are looking at the third type, that is the barley type, which is the characteristic of cereals, we find that stomata rarely open in the daytime. Even if they open in the daytime, during daytime, it is only for 1 or 2 hours, otherwise the stomata are completely closed. So, this is the classification of the stomata based on their opening and the closing patterns. Okay. Now, let us learn about the stomata, what is actually it is made up of. Now, this word stomata is coined from word stomatos. It is a Latin Greek word. Stomatos means a small opening or an orifice or an escaping region. Now, in the plants also we find that they will have a small minute pores which is called as the stoma. Now, this stoma is surrounded by 2 specialized epidermal cells called as the God cells. Yes, once again these God cells may or may not be surrounded by another form of specialized cells called as subsidiary cells. In your anatomy classes, based upon number of subsidiary cells, you might have studied about different kinds of stomata, that is anisocytic stomata, anemocytic type, diacytic, parasitic, tetracytic, cyclocytic. These different types of stomata you might have studied based upon the relation of number of subsidiary cells, whether they are present or absent. If they are present, how many number are there? So, that is one part of the classification. So, here they are covered by 2 specialized God cells. Now, the shape of the God cells is also different. Like if you take in dicotyledons, you find that the shape of the God cells is kidney bean shaped. When if you are considering monocots, especially the grasses, they are usually dumbbell shaped. And these 2 are, as I told you, may or may not be covered by the subsidiary cells, and sometimes the stomata may lead into a small chamber, opening chamber that that goes into the hypodermis of the cortex, that is called as the substomatal chamber. So, stoma with the God cell, subsidiary cells as well as the substomatal chamber all together is referred to as what is known as stomatal apparatus. Okay. Now, as I told you, these besides leaves, we can find stomata on other parts of the plant also, like on flowers you can find them on the fruits also. So, except on the roots you can find stomata at all the places, but the number may be quite varying. That is, you find maximum number of stomata on the leaves, but compared to other parts of the plant. Okay. So, as I have just told you that in dicots you come across the GC, that is the God cells are kidney shaped, and in grasses they are usually dumbbell shaped. Let us look into the nature of this specialized modified epidermal cells called as the God cells. Now, if you look into the nature of that one, you find that the outer walls of the God cells are thin and elastic in nature, whereas the lower inner part of it is quite thick and non-elastic in nature. And because of the thin walls outside, we find that water can move from the neighboring cells, it may be subsidiary cells or normal epidermal cells, water can move from outside into the God cells, leading to what is known as turgidity. When these God cells become turgid, the stomata open. when the water escapes out from the god cells outside because of difference in the osmotic potential, we find that they lose their turgor and the stomata, the god cells become flaccid, and this will lead to closing of stomata. So, in a full turgid god cell, we find that the stomatal pore measures up to 5 to 15 mu microns in width and 10 to 20 mu microns in length. So, we have to remember one thing is that the stomatal pore size is proportional to amount of diffusion that occurs. So, transpiration rate and amount of diffusion of water vapor is quite proportional to the size of the stomatal pore. And you will find that thousands and thousands of molecules of water, carbon dioxide and oxygen can move these from these small pores. That is what about it. Now, let us move further. So, in the picture you can see on your left side you can see a dicotyledonous stomata type, and on your right side the grass type of stomata, and in the center you can see the picture of the stomata, that is the total stomatal apparatus. So, a small opening pore called as the stoma, surrounded by two god cells, and they are surrounded by epidermal cells over here. There are no subsidiary cells in this particular picture. Now, let us move further now. Let us try to learn about other terms like stomatal frequency. Now, what is this stomatal frequency? As I told you, the distribution of the stomata is maximum on the leaves, but you know in nature there are various kinds of leaves, like in mesophyte the leaf nature varies, in hydrophytes it is different, in xerophytes it is different. Even in the mesophytes, if it is going in a tropical area or a temperate area or in an arid region or on mountains, depending upon the environmental conditions, once again the size, structure, shape of the leaf varies. Definitely the stomatal number also definitely varies from plant to plant or from leaf to leaf. You will be surprised to know that within a plant also, in different parts of the plant or on the same leaf at different places, the stomatal number can vary. So, there is one term called as stomatal frequency. Now, what is this stomatal frequency? It is the number of stomata per unit area of the leaf surface. Okay. So, on an average we can find 1000 to 60,000 stomata per square centimeter. You might have definitely done epidermal peeling in your anatomy lab experiments and might have observed the types of stomata. So, you could see that in under a single view you can come across thousands of stomata, you might have observed them. Yes. So, the distribution or stomatal frequency are once again guided by or influenced by external environmental factors also, like light, temperature, humidity. They definitely play a major role in the amount of distribution of stomata in the plants. Now, related to that, let us talk about a few more terms. Now, look at it. Now, depending upon on which part of the leaf the stomata is present, we can classify once again into three categories, that is hypostomatous leaf, amphistomatous type and epistomatous type. Okay. So, hypostomatous condition is such kind of a condition where we find more number of stomata on the lower surface of the leaf, not on the upper surface of leaf. So, in hypostomatous condition, like example being apple, you do not find any stomata on the upper epidermis. You can find them only on the lower epidermis. So, in most of the xerophytes, in plants like ficus, nerium being a true xerophyte, you find the nature of stomata is hypostomatic in nature. Whereas, amphistomatous condition is such kind of a condition where we find stomata equally distributed both on upper surface as well as on the lower surface. And in epistomatous condition, this is characteristically observed in usually in water plants, that is aquatic plants, and we find that the amount of stomata is more on the upper surface and on the lower surface you do not come across any kind of stomata. So, example for amphistomatous condition is most of the herbaceous dicotyledons exhibit this kind of amphistomatous nature. Epistomatous nature, as I told you, is the characteristic of mostly aquatic plants. So, Nymphia, Marsiria are the examples for epistomatous condition. Let us move further now. Now, just it is an extension of what we have studied. So, where in the plant, in the leaf, whether on the upper surface or on the lower surface, and the pattern of distribution. Once again, we can classify the stomata into 5 more types. Okay. So, the first type is apple and mulberry type. Now, what do we see in apple and mulberry type? We find that stomata are seen only on the lower surface and they are completely absent on the upper surface. Whereas, in potato type, we find that stomata are present more on the lower surface compared to the upper surface. Example being potato, tomato, pea. Now, another type is the oat type. Now, in this one we find that there is an equal distribution of stomata. So, equal number of stomata are found both on upper epidermis as well as on the lower epidermis, example being the avena itself. Whereas, in nymphia we find that the stomata are present only on the upper surface, not on the lower surface of the plant, because it being a hydrophyte, that lower part of the leaf is in constantly in the water. Okay. So, the last type is the Potassium type. Now, even this is a hydrophyte plant and is a very interesting condition where we find that the stomata are definitely present over there, but they are vestigial in nature, that is they are non-functioning stomata, characteristically seen in mostly in hydrophytes like Vallisneria and Potassium. Now, presence of vestigial stomata is an advanced feature. If you are taking into consideration taxonomic phylogenetic purview, if you are taking that into consideration, it is one of the advanced features of the hydrophytes, and not only that, it tries to tell that the ancestry of the hydrophytes, that these are derived from the terrestrial plants. So, the hydrophytes evolved from the terrestrial point, and the presence of vestigial stomata is an indication of that kind of ancestry. Besides in the hydrophytes, we do come across vestigial stomata on parasitic plants, on leaves, flowers and fruits, we find this vestigial stomata. Now, since they are non-functional in nature, but what we find is that they can open permanently or close, maybe closed permanently, or they can open and close, but they do not have any function, that is evaporation, that is transpiration does not occur through them. It is a very interesting phenomena. Okay. So, to sum up, what are the main functions of the stomata? Primarily, it is the evaporation, that is loss of water in the form of water vapor into the external environment from the intercellular spaces of the plant. So, the root pressure that has been triggered by the transpiration leads to more absorption of the water along with the essential minerals through the roots from the soil and passes on to the aerial parts of the plant, and this root pressure is constantly maintained because of the rate of the transpiration is constantly maintained during midday. So, whatever amount of water is required, the plant uses them for their metabolic processes, and remaining amount just evaporates outside. Okay. So, primarily it is the transpiration, that primarily it is evaporation. Next one is exchange of gases during respiration and during photosynthesis. And one more thing is that when we talked about the God cells, that is the specialized epidermal cells, now these God cells will have chloroplast in them. This is one of the characteristic feature. So, they can photosynthesize. So, they can produce their own food besides the mesophyll region of the plant leaf. So, we find that, but the number of stomata are quite different. If you look into the God cells, the number of stomata per God cell can be 10 to 15 in number, whereas in a normal mesophyll cell, it ranges from 30 to 70 in number. Okay. Now, let us go further now. Now, if we want to measure how much of transpiration is taking place per plant or specifically one kind of a plant, then there are very different kind of experimental things that we can do to measure the transpiration. But before that, we will just look into the different terminologies. Okay. So, if we are trying to count the rate of transpiration per plant, then it is defined as grams of water vapor per second per plant that has moved out of the plant. That will tell you about the rate of the transpiration for a plant. If we are talking about rate of transpiration occurring at a leaf level, then it is defined in the form of transpiration flux. So, what is this transpiration flux? The quantity of water vapor transpired by unit area over a leaf surface in a unit time. It is measured in grams per hectare. If you are taking a large crop field into consideration, then we are going to take it into kilograms per hectare, then counting it. Okay. Now, this diffusion of gases from outside intercellular spaces of the plant to external medium is because of the water vapor pressure gradients that generated between the intercellular spaces of the leaf. Now, because of the change in this gradient, movement of water vapor occurs inside to outside environment. Okay. Now, let us discuss a few more points of it. Now, as I told you in the previous slide itself, the rate of diffusion is proportional to the stomatal pore area, that is circumference. What is the width of it? Depending on that, the rate of the diffusions also vary. Okay. So, though the stomata are present evenly distributed on the leaf surface, again depending upon the kind of plants, that is what epistomatas or amphi or hypostomatas, whatever condition, the total number of the stoma, if we pool them together, it amounts up only to 1% of the total area of the leaf. But you find that 50% of evaporation compared to land area, compared to a normal water area, the rate of transpiration through this minute pores is more than 50%. So, as I told you earlier, 15% of the total atmospheric water is contributed by this process called as evapotranspiration. So, these minute pores can do such large amount of water contribution to the atmosphere. Yes, not only that, carbon dioxide percentage in the atmosphere we know is quite less, it is only 0.03%. Okay. So, when the rate of carbon dioxide in the internal medium of the leaf is less, it facilitates movement of carbon dioxide from the atmosphere into the leaf medium through the stomata. Okay. And carbon dioxide is quite essential because we know that photosynthesis is related to the carbon dioxide absorption and its fixation, isn't it or not? Yes. Now, let us look into mechanism of stomatal movement. It is only just revising what we have discussed in the previous slide. So, the guard cells are the ones which open and close, and when they open the stoma is seen, and when they close the stoma does not appear. So, the movement is correlated with photosynthesis. So, during photosynthesis, as I told you, even guard cells, since they have chloroplast in them, they can photosynthesize. So, when the sugars are produced, we find that the osmotic potential changes. So, more amount of negative osmotic potential leads to endosmosis, that the water enters from external medium, that is surrounding subsidiary cells or surrounding epidermal cells into the guard cell, making it turgid type. Yes. So, since the outer walls of guard cells are quite thin, they open up, leading to pulling of it. So, the inside of the guard cells, since they are thick in nature, non-elastic in nature, they exert some kind of a pressure leading to opening of the stomata, and the reverse occur when photosynthesis goes down. Okay. So, photosynthesis is related to stomatal opening and closing, and the opening and the closing of the stomata is governed by guard cells. Okay. So, that is what we have put on this slide also. So, when the guard cells become turgid, they absorb the water, and the stomata opens, and when they lose the water, they become flaccid and the stomata closes. Okay. So, this stomatal movement in the guard cells is regulated by the osmotic potential, or what we call it as the turgor pressure. This is as I told you, is because of the movement of water from outside medium to into the guard cells, or the movement of guard cells once again going outside. Okay. So, that is what is about the process of opening and closing of the stomata. Now, there are various theories trying to tell how actually these stomatal movements occur, at what point of time they occur, because every plant has its own internal biological clock, and not only that, this biological clock is adjusted to the diurnal pattern, that is sun rising and sun setting time also. So, major physiological processes also go side by side based on these diurnal patterns. So, there are different theories trying to explain about this opening and closure of the stomata and they have put their own interpretation, but most of these theories are closely associated with photosynthesis. So, you can see on the slide there are 5 theories, that is von Mohls theory, starch sugar hypothesis proposed by Lloyd, Stewards scheme, this is an extension of starch and sugar hypothesis, then we have glycolate mechanism, and then theory of potassium ion transport proposed by Leavitt. Now, we will try to elaborate on them. Now, look into the von Mohls theory. Now, von Mohls has proposed that when during daytime, when the light intensity is starting to go up, besides the light, even the temperature will also start increasing. That is, light intensity increases, temperature also increases. So, because of the light intensity, photosynthesis occurs in the plants, especially in the leaves and in the mesophyll region, along with the mesophyll region in the god cells also. Yes, and because of the photosynthesis, the end product we know are the sugars, that is the glucose, yes, which is then converted into starch during the night time, we all know it, for storage. Yes, so formation of sugars occur. Because of the formation of sugars occur, there is a difference in the water potential. It leads to endosmosis, that is movement of water from outside to inside the god cells, that is movement of water from hypotonic region to hypertonic region. Yes. Now, as the water enters through the into the god cells, we find that there is an increase in the turgor pressure. This will lead to opening of the stomata. The reverse goes when the water moves out and the closure of the stomata occurs. This is the concept of von Mohls trying to explain the stomatal movements. Now, let us look into the next theory, that is sugar starch hypothesis by Lloyd. Now, according to Lloyd, what he inferred that the god cells will have starch stored in them during night time, and during daytime these god cells will again break them down into sugars. So, once the sugars are produced, the same thing what we have discussed in von Mohls theory occurs one more time. Because of accumulation of sugars, the endosmosis occurs. Because of endosmosis, the turgor pressure of the god cells increases, and because of increase in the turgor pressure, these god cells become turgid and leading to opening of the stomata. That is what you can see, and during the night time the opposite occurs. One more contention over here is that if the god cells are storing starch during night, we find that the adjoining mesophyll cells store the starch during the daytime. So, this shift interconversion between the starch and sugar leads to the stomatal movement. Okay. Now, let us move to the third theory, that is the Stewards scheme. Now, what does Steward tries to tell you is that when the starch is converted into glucose 1-phosphate, this glucose 1-phosphate is not able to not sufficient enough for the stomata to open. So, what happens is that this glucose 1-phosphate is converted into glucose 6-phosphate, and this glucose 6-phosphate leads to opening of the stomata. Once the stomata are opened and after a period of time, once again it gets breaks down into glucose and inorganic phosphate. Okay. So, this one leads to stomatal opening. Okay. After a period of time, that is after sunset or when the stomata are closing out, what happens is that this glucose is once again converted into glucose 1-phosphate because of the hexenokinase enzyme and also because of availability of ATP. So, starch is converted into glucose 1-phosphate. This glucose 1-phosphate is not sufficient for stomatal opening. So, it is converted into glucose 6-phosphate. Glucose 6-phosphate is once again broken down into glucose and inorganic phosphate by their concerned enzymes. Now, when this glucose is formed, it leads to stomatal opening. Once the glucose is once again reconverted to glucose 1-phosphate because of hexenokinase enzyme and ATP, the stomata close. This is what is called as the Stewards scheme. Okay. The other picture is also related to what we have just discussed, that is about the Stewards scheme. You can see on one side stomata being open. How does they open? Only when the glucose 1-phosphate is broken down to glucose. Yes, and the stomata close once again when the glucose is reconverted into glucose 1-phosphate. Now, let us go further now. Now, let us look into the another related theory called as the glycolate mechanism. Now, this was proposed by Zeltich, and he said that when the concentration of carbon dioxide is less in the plants, that is in the leaves, what happens is that in the god cells, the glycolate is oxidized into glyoxylate, and this glyoxylate is further converted into sugars along with ATP that is being produced. Now, what happens is that once the sugars are produced, water moves into the god cells due to end osmosis and the stomata open. This is what is proposed by Zeltich, and his theory is known as the glycolate mechanism. Now, let us look into the last theory and most followed theory, that is the potassium ion transport. Excuse me. So, what do we find is that there is something called as potassium ion pump. Theory called as potassium ion pump. Now, what do we find in this potassium ion pump is that when the god cells become turgid, we find that H plus ion, that is hydroproton ions, move outside the god cells and the potassium ions come inside the god cells, and these are balanced by anion cation balance occurs over there because of organic compounds as well as the chloride ions. They maintain that electronic balance, leading to turgidity and opening of the stomata. Now, what is this is known as the potassium ion pump. So, H plus extrusion occurs and the hydrogen ions go outside and potassium ions come inside and leads to opening of the god cells. Now, among the 5 theories what we have studied, among them the potassium ion concept is the most approved concept if you are talking about the stomatal movements. Now, let us look into what factors will affect the stomatal movements. As I told you, the stomata are quite sensitive to environmental changes. Now, light, temperature, humidity, all of them have definite bearing on them, on opening and closing of them. Not only that, the endogenous signals that occur within them, like amount of abscisic acid or amount of availability of water, all such kind of internal factors also will have very prominent effect on opening and closing on it, and definitely the osmotic concentration also plays a major role in opening and closing of the stomata. Now, let us look into the external factor, the light. The quality and quantity of the light is quite important. You know that in the wavelength visible spectrum, among the other spectra, the most favored light for the stomata to open is the blue light. Of course, red light also favors it. If infrared and UV rays are there, stomata will not open. Why the blue light favors is that this blue light links with the potassium ion transport channel. Not only that, we know that light is the essential feature for the photosynthesis to occur in the mesophyll cells as well as in the god cells. Hence, it acts as a very predominant role in stomatal control. Okay. So, along with the light, other factors may help in stomatal opening and closing. The other factor is carbon dioxide concentration. Now, this carbon dioxide concentration has a very deep impact on opening and closing of the stomata. So, less concentration of carbon dioxide leads to stomatal opening, now more concentration leads to stomatal closure. So, in the daytime usually we find that the stomatal concentration is quite less, because whatever the CO2 enters into the life, immediately it is used up for photosynthesis. But when the sun sets, we find that the concentration of CO2 increases in the plants, especially in the leaves, because of respiration. Now, more accumulation of the carbon dioxide ultimately leads to immediate closure of the stomata. Okay. Now, the effect of the minerals. We have just discussed that opening of the god cells are guarded by the potassium chloride and hydrogen and organic acids. So, not more elaborating on that one. So, we will move to the next slide. Now, this is one important thing. You might have observed that plants usually show a bit of wilting during hot summers. When the afternoons are in peak, you find that the leaves wilt. Why do you think this is? This is because of loss what is called as the water stress. Now, this water stress is triggered by the endogenous signal, that is the internal phytohormone, that is the abscisic acid. We know that abscisic acid is a inhibitory phytohormone. Yes. So, when the concentration of ABA increases, that is from the mesophyll it enters into the guard cell, we find that it leads to water stress, and the entry of the abscisic acid into the guard cells leads to closure of the stomata. Once again, the abscisic acid entry, its process is once again interrelated with the carbon dioxide also. Not only that, one last point I would like to add over here in abscisic acid is the calcium ions. Even the calcium ion concentration, which are usually present in the vacuoles or in the cytoplasm or between the spaces of the cells, that is in the apoplast, when they move into the guard cells, they also lead to closure of the stomata. Okay. So, this is the mechanism of stomatal opening and closing, what we have discussed in all the previous slide, the same thing. So, during day what happens, during night what happens. One thing I would like to say that it is nothing but it is purely related to photosynthesis, yes, in the guard cells and the mesophyll, purely related to difference in the osmotic potential that leads to either opening or the closing of the stomata. Now, this is all about the mesophytes or the general plants. Now, what happens in plants, one kind of xerophytic plants that are the succulents. Now, in succulents we find that stomata never open during the daytime. They open only in the night because at that time the carbon fixation occurs. So, they follow the C4 pathway. They usually, we call them as camp plants also. Example being lot of water storing xerophytes like cactus, aloe, calonche, sedum, etc., etc. Yes. Now, let us talk about the stomatal movements occurring in them. Now, in them the kind of stomata which open in the night, that kind of stomata are called as scotoactive because they open only in the night, okay, and close during the day. Okay. So, what happens is that CO2 fixation occurs in the night, and due to that acidification, CO2 leads to lowering of CO2, and usually since the concentration of the carbon dioxide becomes less, stomata open. But in the daytime what happens is that deacidification leads to decarboxylation leading to stomatal closure. Let us move further. Now, what are the factors that affect the rate of transpiration? So, if you are talking about the rates of transpiration, both external as well as internal factors play a major role. If you are talking about the external factors, humidity, temperature, light, wind, water supply and atmospheric pressure play a major role. So, what do we find is that humidity, if you are talking about humidity, it is about the wetness or the coldness of a particular place. So, we find that in arid regions, in dry regions, more number of stomata are present in that one. So, more amount of transpiration occurs. So, but when we look into wet areas where like Amazon areas or where plants are very close to the water, we find that the rate of transpiration is quite low in them. So, in humid conditions, we find that the transpiration rate becomes less, whereas in arid or dry conditions we find that the rate of transpiration increases. So, interrelated with this, temperature and light are two interrelating factors we have already discussed. So, temperature also at 0 degrees temperature we find that stomata close, but as the temperature increases we find that the stomata also open. So, the optimum temperature till where the stomata remains open is somewhere 30 to 35 degrees temperature. Okay. So, maximum optimum temperature at which the stomata remains open is 35 degrees. Beyond that we find that the stomata close. So, high temperatures and a very low temperature leads to closure of the stomata. So, light, the importance of it we have already discussed. Now, coming to the wind, a gentle breeze will lead to increase in rate of transpiration. Why? Because we find that at the surface of the leaf we called as what is known as boundary layer. Now, what is this boundary layer? It is a moist wet area over the surface of the leaf which facilitates escape of water into the form of water vapor outside the leaf. Now, this boundary layer is a very delicate layer. So, if it is a gentle breeze, it does not affect much, but in high voltage winds we find that this boundary layer is completely destroyed and it leads to changes in the transpiration rate. Now, water supply, we know that without water transpiration does not occur at all, isn't it or not? So, depending upon the water supply, the rate of transpiration can increase or decrease. Yes, and atmospheric pressure, if the height goes on increasing. If it is a particular plant which height is going up constantly, we find that as the plant reaches greater heights, we find that the rate of transpiration goes down. But if we are talking about plants in high altitude areas, we find that since the temperature is low, we find that the transpiration rate also comes down. Okay. So, increased altitude to some extent increases the rate of transpiration, but plants living in a higher altitude, because of low temperature, we find that the rate of transpiration falls off. Okay. Then let us talk about the internal factors also. Now, if we are talking about internal factors, leaf area, orientation, leaf size and its shape, leaf structure, root shoot ratio play a prominent role in deciding the rate of transpiration. So, if you are looking into leaf area or orientation, what we find is that a large leaf will have widely spaced stomata. So, less number of stomata widely placed. When we are talking about a narrow leaf, it has a smaller number of stomata aggregated together. So, rates of transpiration will definitely differ. Not only that, the incident of light, if the leaves are perpendicular to the incident of light, then the rate of transpiration goes up. Okay. So, smaller leaves, if you are talking about the size and shape of the leaf, smaller leaves, dissected leaves show more rate of transpiration because they have more amount of vapor caps on them. Boundary layers are present in more in number, hence we find that the rate of transpiration also increases in smaller leaves compared to the broader leaves. Okay. And the leaf structure also, if you observe a xerophytic plant, you find that they usually have a very thick cuticle. Epidermis also, you find that stomata are also sunken in nature. You find modifications according to the xeromorphic conditions that are over there. So, if you compare it with a normal leaf structure, it is quite different. So, depending upon the type of the leaf also, the rate of the transpiration varies. Okay. So, higher root shoot ratio also leads to more rates of transpiration. Okay. Now, let us talk about antitranspirant. Now, in the beginning slide I said that transpiration is a necessary evil which no one can control, but it is an important physiological process also, so that water balance maintains in the plant, isn't it or not? But lots amount of transpiration can be quite determinantal to useful commercial plants. So, what can be done? This can be done by applying externally what is known as anti-transpirants. Now, this anti-transpirant could be a normal phytohormone, that is ABA, or some other kind of chemicals which are applied in very less concentration can lower the rate of transpiration. Now, these phenyl mercuric acetate what we are seeing over there, they are usually not harmful. Very conveniently for commercial purposes, these can be used to reduce the rate of transpiration. So, besides that, silicone oils, waxes, colorless plastics can also be used as anti-transpirant. And of course, if in the condition we have less amount of carbon dioxide, like in greenhouses, we have controlling system is there. So, less rate of CO2 also will inhibit the or try to control the rate of transpiration. Now, this was all about transpiration. Now, transpiration we have understood that it is expelling of water, extra water from the plant to outer source in the form of water vapor. Now, there are two more physiological processes that we can find in few group of plants. One is guttation and one is exudation. Now, let us try to understand them. Now, what is guttation? Early morning when you are walking by a garden or a plant, you might be seeing that water droplets accumulate in its leaf margin areas. So, loss of water in the form of liquid, not water vapor, in the form of liquid from the uninjured margins of the leaf is called as guttation. Now, in this guttation, what comes out is pure water. Now, what comes out, it is called as guttation fluid also. Now, this guttation fluid will constitute along with water dissolved solutions like different kinds of sugars, enzymes, amino acids, even vitamins and minerals are also found in the guttation fluid. And how does this guttation occurs? It occurs by special pores called as the water pores, or they are called as the hydrodes. Now, this guttation is caused by the root pressure. Now, look into the electron microscope picture of an hydrothode. So, what you find is that each hydrothode or the water pore will have a small opening called as the water pore on its tip region, followed by a large air chamber. Now, this large air chamber is followed by a loose mass of thin parenchymatous tissue, which we call it as the epithem. Now, this epithem will take the midvein openings open into, that is the tracheids of the xylem or open into this epithem. So, due to root pressure, when lot of amount is taken up by the leaf, excess amount gets collected in this air chamber because of the vein giving out the extra water. Now, in the air chamber, this water gets collected, and during humid conditions, that is in the late night or early mornings, you find the exudation coming out in the form of small droplets of water. Now, let us look into the next process called as exudation or the bleeding. Now, guttation was all about oozing of liquid, that is aqueous liquid, water plus minerals and others from uninjured part. But if you look into few family members, like you look into Asclepiadaceae member, Moraceae member, when you try to pinch a tip region of a leaf or a stem, you find that latex coming out of it. That is oozing of liquid from injured incised parts of the plant is called as exudation. Now, this exudation comes from either xylem, phloem or from the cortex. Even the reason for exudation is the root pressure. Examples are given over here: milky latex that comes from euphorbia. The best example is the rubber latex. Yes. So, you find phloem exudates in agaves and yucca also. Now, any theoretical learning is not sufficient if it is not followed by lab activity. So, if you want to learn the process in a laboratory also, you can learn through this processes. So, you can use you can learn transpiration, guttation and exudation doing a small lab activity. That is just to tie up a polythene cover during early morning to a few group of leaves, tie them up tightly and observe after the sunset. You will find that due to transpiration, water after water vapor, through the water vapor it condenses back to water and gets accumulated in the polythene bag at it one. So, if you want to observe guttation in the mornings, you can observe them. If you want to observe exudation, you can just pinch any part of these plants which gives out the exudate to observe it. Now, lab activity, as we are going for online classes, if you want to learn lab activity virtually also, there are so many platforms online to learn virtual experiment, like Amrita Technologies is there, Pixar is there, yes, VLabs are there, where you can learn all these experiments online too. Okay. So, before I wind off, I would like to give few reference books and websites for your PG entrances, especially if you are talking about the physiology. So, Plant Physiology by Sinha and Pandey, textbook of Plant Physiology by Verma, Plant Physiology by Ross and Salisbury, Fundamentals of Plant Physiology by VKJ are one of the most popular and in demand books for physiology preparation, and textbook of Plant Physiology by Verma and Ross and Salisbury are like Bible for any physiologist. You can get all the required information from the basic level to highest research level compiled in this book. So, it is a must book for any physiology loving person. Now, you can see the websites over here where you can practice, as I told you in the previous session also, you can practice the mock sessions for your PG entrances. So, Toppers exam is one website, Exam Race is another one, Biotechnica is there, Easy Biology is there, through which very easily, freely you can practice your MCQs and prepare yourself online sitting at the comfort of your home for your PG entrances. So, with this I come to end of our session in plant transpiration. I generously thank Gurukulam. I generously thank our dynamic secretary sir, R.S. Praveen Kumar sir, and all the higher officials associated with our RDC for giving me an opportunity to come before you. So, stay safe, stay focused, keep learning. Thank you then.